Good afternoon, everyone. I hope you can hear me and I guess see my slides. So I would again like to thank the organizers for the kind invite. I'm really looking forward to talking to you about CAR T cell therapy role in ALL. These are my financial disclosures. So I have split my talk. First, we'll talk about BLL, and then towards the end, for a few minutes, we'll talk about TLL. And I've also tried to show some of the data from ASH meeting, some new updates on the data from the ASH meeting. So first, let's talk about BLL, which is obviously the largest of the disease group. So we already have some agents approved in relapsed refractory BLL, namely Inotuzumab and Blinatumomab. These were the two studies in New England Journal led by Dr. Kantarjian here, which really established and led to the approval of these drugs. And I think, as you can see here, these lead to a median OS of about 7 months for both Blinatumomab and Inotuzumab cohort. But now we have several CAR T products which are approved in the United States. And Dr. Majel already kind of alluded to many of these. And just to kind of highlight these products, there are 3 currently approved products, or approved constructs, I should say, in the United States. And they differ slightly in terms of their co-stimulatory domain. For example, the Gilead product, or the previously Kite product, had a CD28 co-stimulatory domain. And the Novartis and the BMS product have a 4-1BB. Now, as I mentioned, these 3 are currently FDA approved. And the 2 specific which I've highlighted here are the ones which are approved for adult BLL. So namely, CTL019, Tisagenlecel, is approved for children and young adults up to the age of 25 years. And more recently, with the approval of Brexel, which is the Kite product for adults with relapsed refractory BLL. So we'll talk about these 2 in more detail about the data which has been so far published for these 2 drugs. So first, the Ileana trial. Actually, this was last updated now some years ago, 2018. We haven't, or at least I haven't seen a further update of this data. But this is the trial which led to the approval of the product called Kymriah. Trade name is Kymriah, or Novartis CTL019. So this was a trial which was designed for children and young adults up to the age, actually eventually the age was up to 25 years for these patients. And they had to have relapsed refractory disease with 5% or more lymphoblast. And again, this product I just kind of highlighted here, this is a 4-1BB construct. The reason I'm alluding to that is there's a difference between 4-1BB construct and CD28 construct. So when you have a 4-1BB construct such as this product, you expect lesser peak of the CAR and you expect more persistence of the CAR. For products such as Kite product, which is a CD28-based product, there is a higher peak of the CAR expansion, but then they don't tend to persist that long. And I will show you some data to that effect. So this is a trial where they used fludarabine cyclophosphamide as lymphodepletion, which is a pretty standard lymphodepletion chemotherapy across CAR T products. They overall screened 113 patients. Finally, they infused 79 patients. Some patients had issues with manufacturing, or they died or had issues after getting enrolled prior to getting the cells. So among the 79 patients, you can see this is a largely a pediatric study, and quite a few patients, 61% had prior stem cell transplant, and 3 median lines of therapy. So a refractory group of patients who were treated in this study. Now, Dr. Majil talked about the toxicities, or in general. So again, that's something very important for any of the CAR T program, to develop a unified strategy of how you're going to manage the toxicities. And the reason it's important is that if you look at this particular study, almost half of the patients had grade 3 or 4 toxicities seen. So again, that's cytokine release syndrome seen, sorry. So again, that's something you need to know how to manage those patients. And at least in this study, half of these patients required ICU admission, and then 1/4 of these patients required high doses of vasopressors to again maintain their blood pressure. So again, it's something which needs coordination and care, not just with the leukemia or the transplant team or lymphoma team for that matter, but also ICU, critical care, neurology, and other disciplines. Now, the strategies tend to work very well. So, for example, in this 79 patients, 65%, 65 of 79, so 82% achieved CR/CRI, and practically every one of them had MRD negative remission. And you can also see that there appears to be a durability of these remission. But as I said, that this data, I believe, was last updated in 2018. So I'm actually myself curious to see an update of this data, because certainly we will have at least additional 2 or 3 years of follow-up to this curve. So this is the Novartis product, which is currently FDA approved in the United States. Now, at this ASH meeting, the Novartis team showed data about a new product, which they call it as YPB323. So the CAR construct is actually exactly the same as Kymriah. It is a CD19 CAR, autologous CAR, and it's with 4-1BB construct, 4-1BB costimulatory domain. But they have developed a proprietary manufacturing process, which is only what they say is less than 2 days. And therefore, this prevents ex vivo T cell exhaustion, and they call it T-charge platform. And in fact, the data they showed at ASH in non-Hodgkin lymphoma setting show that with 25-fold lower doses of CAR T, they were seeing the same expansion as their T-cell product, which is the Kymriah product. So I'm not going to go over the data in more detail, but I just want to show you this was the presentation with Dr. Ian Flynn at ASH, where they showed clinical data, and also there was a preclinical data poster as well, that you can use this T-charge platform. Again, this is a Novartis proprietary platform for these patients. And what they showed is that, again, with 25-fold lower doses, you're seeing the same expansion and less exhausted T cells. So I think it will be interesting to see. I think this product is now, this particular trial is also open for relapse refractory BLL. So I think it will be interesting to see how this pans out in future. Now, that was a product by the Novartis, which is approved for children and young adults. More recently, last year, we had, at least in the United States, we had approval of Kite product called Brexel, which was based on this trial, which was published in Lancet. So this is called a ZUMA-3 study. And again, just to highlight, this is a CD28-based product. And this was an autologous CD19 CAR for adults 18 years or older, again, with 5% or more marrow blasts. So here, 55 patients were treated. Again, the lymphodepletion, which is pretty standard, fludarabine, cyclophosphamide, and the approved dose is 1 million CAR T cells per kilogram. Median age was 40 years, so older patients, 2 prior lines of therapy. Now, again, you can see that these patients were heavily treated. Half of these patients had Blina, about half had transplant, and some patients had prior Inotuzumab. And again, the median pre-lymphodepletion bone marrow blast count was 59%. So these are patients with quite active disease, which received the CAR T cell therapy. And again, 2 prior lines of therapy and multiple of these standard approved therapies patients had received. So when you look at the 55 patients who were treated, 39 achieved a remission, and 30 of the 39 achieved MRD negative. So practically everyone who achieved a remission achieved MRD negativity. So overall, I think it's fair to say 70% MRD negative remission with this product. So the next question comes up is, what is the duration of remission? How durable these remissions are? So you can see here, for patients achieving a remission CR/CRI, the median remission duration was just over a year. And then if you look at the overall survival for all treated patients, it was about 18 months. So that is quite interesting, because early on, I showed you a data slide with Inotuzumab and Blinatumomab, where in the relapse refractory setting, the median survival was 7 months. So certainly here, we're seeing a median OS of 18 months, which certainly looks encouraging for this patient population. Now, I think, again, as we talked about in the pediatric study with Kymriah, you have to see what are the toxicities. So cytokine release syndrome median time to onset was 5 days, and 1/4 of the patients who had it. ICANS, median time of onset 9 days, again, 1/4 of the patients had it. And the one patient did die from brain herniation, making a point that these are therapies which can potentially lead to significant complications. And then because of these rates of ICANS and CRS, ICANS, yes, you can see tocilizumab, steroids, vasopressors, and quite high number of patients were given. Now, it's kind of, I think, pretty intuitive that if you have higher CAR expansion, more likely to achieve complete remission, more likely to achieve MRD negative remission, and some correlation with cytokine release syndrome as well as neurotoxicity. So if you don't expand the CARs, you're not going to have toxicities as well as efficacy. Now, but one question which comes up many times in the context is that, do you need persistence of these CARs for a response to occur? And I think, as I mentioned before, I think there's a difference between the constructs. For example, for the Kymriah, which I showed you before, there is good data that you need persistence of these CARs. If you don't have persistence, or if your B cells, your normal B cells recover in the peripheral blood, high risk of relapse. But for Kite product, the CD28 product, and again, this is from the paper itself, at 6 months and at 12 months, very few patients who were responding in ongoing response had persistence of the CAR. And in fact, at 12 months, all 10 patients who had in remission at that time had recovered their normal B cells. So kind of somewhat opposite of what we see with other CAR products. And again, as is true with CAR, 1/3 of the relapses were CD19 relapses in this study. So I think, so I just want to kind of give you a flavor of the approved CAR T product in the United States, and especially with the approval of this Kite product, because that spans all adult relapse refractory BLL. Our center and many other centers are now starting to treat patients with this standard of care CAR in relapse refractory BLL setting. Now I want to quickly move to some new data or some, I guess, upcoming strategies. So what we talked about is the CD19 CAR. But what about dual CARs? Why can't you have 1920, 1922? And those are the strategies which are ongoing right now. So when you start talking about dual CARs, there are different ways you can have a dual CAR strategy. You can infuse two products together. You can co-transduce them. So some products will have one CAR, some products will have two CARs, some products will have just one CAR. You can have one vector, which makes two different CAR constructs, or you can have one vector, which makes two different CAR constructs on the same one construct. So there are different ways you can construct these CARs. And there are pros and cons of these approaches. As far as I can tell, there's no right or wrong, or at least there is no best strategy we know of. Now, this is a slide I made after ASH meeting to kind of review what's happening in the field. And this is only focused on B-ALL, non-Hodgkin's lymphoma. There are other CARs happening, but this is only focused on B-ALL and looking at dual 1922 CAR. I won't go into detail much, but there are, suffice to say, there are several published data set. The first 3 are published, and the last 3 were at the ASH meeting, where different strategies, different constructs are being used to make dual CARs. And I thought I would just highlight one briefly, where from UPenn group, Noel Frey, where they did co-administration of the CAR. So what they did was they made 2 separate CAR products, CD19 and 22, and they infused them in a graded manner, 10% on day 1, 30% on day 2, or the next day, and then subsequently 60%. And this has been their strategy. So these are 2 different CAR products they infused. 13 patients were treated, again, heavily pretreated patients. And again, with the caveat of small number of patients, anything, these 11 out of 13 patients have responded, and most of them are in ongoing remission at this time. So I think, whether you need a dual CAR, obviously that remains to be seen. But I think very briefly with the data I showed you, I think I hope you can still hear me. Hello?
Yeah, so we have a slide, allogeneic CARs, is that right?
Okay, Sure. Okay. Some of the screen on my side is somewhat frozen, so I was not sure. Okay. So I think before I go to the allogeneic CAR, yeah, so I think the jury is out whether you need dual CARs or not. And there are also now concepts of triple CAR that combining 19, 20, 22 all together for the patients. And again, those trials are just starting right now for patients. Now, Dr. Majil had mentioned about allogeneic CAR as well. So obviously we know the autologous CAR. This is the kind of what the approved products are. You have apheresis product from the patient, you make the CARs out of them, and then you give it back to the patient. Now, what is an allogeneic CAR? An allogeneic CAR, as kind of the word implies, you're deriving the CARs, the immune product, the T cells or NK cells for that matter, from healthy donors or cell lines or from umbilical cord. So these cells are already pre-made because they're derived from healthy donors. You can already pre-made them, do the QC analysis. So these CARs are like drugs available off the shelf in your lab, which you can infuse anytime a patient is there. You need to do the CAR. So again, there's an advantage and disadvantages of these allogeneic CARs. So I made this table just to kind of very briefly summarize what are the difference between autologous and allogeneic CARs are. Certainly what we heard today and also Dr. Majil, majority of the things he mentioned was an autologous space where we have clear approved efficacy, persistence, but then the issue is manufacturing, use of bridging chemotherapy. Certainly in the context of ALL, if you have a white count of 0.1 for your ALL patient, you're not able to collect T cells from those patients to make an autologous CAR. Allogeneic CAR, have all those advantages in them, but the issue so far is that we have very limited clinical data. There's an issue of persistence because they don't last long, and there's a potential risk of graft versus host disease because you're infusing someone else's CAR T cells. So we reported this data in an oral presentation at ASH about an allogeneic CAR product called PBCAR0191 in relapsed refractory adult PLL. I thought I will briefly share with you. Again, these are derived from healthy donor T cells where they have a knockout of T cell receptor to prevent graft versus host disease, and then a CD19 CAR is inserted. We investigated 2 different dose levels, 2 different lymphodepletion regimen, so a standard lymphodepletion and an enhanced lymphodepletion where the doses of cyclophosphamide was double and an extra day of fludarabine was given. Now, 15 adult patients were treated in the study with a median age of 46 years. As you can see, 45% with the median pre-lymphodepletion blast count and 5 prior lines of therapy. Now, one thing we noticed in this study was that for these CARs to persist longer or to persist at a higher Cmax, if you either increase the dose of the CAR or you increase the depth of lymphodepletion, you're able to increase almost 100-folds the persistence or the area under the curve of these CARs as compared to using standard lymphodepletion with standard doses of these CARs. So I think you can improve your CAR efficacy in the allogeneic space by modulating the CAR cell dose or by lymphodepletion. Now, with this caveat a small number of patients, among the 9 patients who either received enhanced lymphodepletion or a higher dose level, as you can see, we see 5 of these 9 patients that achieved MRD negative remission. So again, this is a field which is slowly evolving. Right now, there are many other clinical trials in the allogeneic space happening, not just with this product, obviously with many other products, and it remains to be seen how they will pan out. Now, very quickly in the last few minutes, I just want to touch base on TLL, which is something of my own research interest as well. And for TLL, I think that the challenges have been there, and we don't have any approved product for this, for TLL. And the issue, there are some challenges issues, which are T cell aplasia, because you can deplete your T cells if you give a CAR, and then it can increase the risk of infection. There's issue of fratricide, where because the T cells, your CAR T cells express the same protein, for example, CD7, which you are trying to kill on the tumor cell, so the CAR T cells can kill each other. Or you can contaminate your product because you're trying to isolate T cells from a pool of mix of T cells and T cell lymphoblast, and you can transduce a T lymphoblast with your CAR. So there are strategies to mitigate all these things which are ongoing right now. And some of these, you may have seen it, they were published in JCO and some EHA last year. This is a study from China where they use 4-1BB co-stimulatory domain to treat patients. This is from the paper in JCO where, again, they did it in the context of a transplant. So the T cells are derived from donor-derived T cells from the transplant donor targeting CD7. And I think just from the big picture view, you can see most of these patients are in remission, in ongoing remission at the time of this publication. Another group in China is developing another CD7 CAR. Small number of patients were treated, but again, with good activity so far. And again, this was reported in Clinical Cancer Research recently. And then from the ASH meeting, another group from China actually reported, this was from the ASH presentation itself, high effectiveness and safety of, again, a CD7 CAR. And I will show you one data slide, just in the interest of time. You can see they treated about maybe 12, 13 patients here. Most of these patients are in remission, in ongoing remission at the time of this analysis. So I think
2 minutes, please.
The point I'm trying to make is that these strategies continue to evolve, and I think hopefully we'll start seeing more US-based or other institution CD7 CARs and other targets for this disease. In the last 2 minutes, Dr. Majel mentioned about the CAR T toxicity, so I think I just want to make sure everyone is aware that there is a consensus criteria established by ASTCT where you can grade your cytokine release syndrome based on different, these are set criteria now established and all the clinical trials use it based on fever, hypertension, hypoxia. There is a specific criteria for ICANS, which is the other name for neurotoxicity. This is a ICE score, which is a part of the ICANS criteria, and just want to specify what this is. So again, this is very important, again, in your practice, at least on a day-to-day basis when we're seeing managing patients with CAR T. My last slide, so I think I showed you some data. There are 2 approved CD19 products in the US, and those are listed here. I think that what's happening in the field in terms of the clinical trials for our group and other groups is trying to introduce this in earlier lines of therapy. Why do you have to wait till salvage 3 or 4? Why can't you do it in salvage 1 in high-risk patients? Or maybe even as an MRD, you consolidate the patients with few cycles of chemotherapy and then do it as in MRD positive or even MRD negative state as a consolidation. Several of the dual CARs and also not triple CARs in development. We'll see how that pans out. Allogeneic CARs have pros and cons, but again, several ongoing clinical trials, still, I think, early phase of development for these CARs. And then TLL, I think several CD7, there's also CD5, there's some other targets being explored as well. And that is, I think, an area of really research and unmet medical need. Thank you for your time and attention, and happy to take any questions.